Hi, this is Sergei from 100x Bio, and in this video, I want to share with you what were my personal pains, which this startup which I'm building is solving. So, what problems of a T-cell immunologist 100x Bio can solve? One of the big problems is the protocols people work with in the lab are extremely long and error sensitive. And I remember myself when I just started my career as a graduate student uh, or working as a postdoc. I remember how much stress it is working with some expensive reagents. Sometimes your kit costs like half of your monthly salary, and you know that you can't lose the reagent just for nothing. You know that you don't have a second attempt if you make a mistake, or maybe you have a second attempt, but uh, even if you have a second attempt, you can lose a work of several days, which you've been doing before, and this develops a lot of anxiety around the experiment. It develops a lot of stress, and very often, the processes and protocols are so long that PhDs or postdocs they work long hours. So I remember myself having 16-hour long workday or 14-hour long workday, and there is no way around because these processes are very manual. They are not even half automated, and it involves pipetting. It involves very attentive control of tiny little drops which you're working with, and sometimes you need to control them over hours and hours. And of course, it's unavoidable making some mistakes. And whenever you make a mistake, there are different options. Sometimes you know how to recover from this mistake. Sometimes you need to recover right away, fix something, and then uh, imagine that never happened. Uh, of course, you will be thinking like, now, even if I recovered the mistake, this can probably impact the data. So when we get the data, we can think, looks like nothing happened. Looks like all the numbers look straight, all the numbers look correct. But sometimes, even if you recovered this mistake, when the data came out, you will be like, okay, this point seems to be quite off. And then it becomes subjective. Do we still want to include this data? You want to remove this data? And many good scientists would have different opinions on this, like how to handle those tiny situations. There are other situations when you can recover the data with a limited time frame to do the error recovery, and if it took too long, like it's impossible. So you're not only in the stress because you made a mistake, you're also in a hurry, and sometimes you're in a hurry just by the way the process is organized. You have to do some operations in seconds, and if something is wrong, like you used all of the tips for your pipette, and now you need to make the next operation in a few seconds, and you don't have a new box of the pipettes, you need to go search for the new box within seconds, and you need to open it, you need to restart, and again, there will be this feel like, okay, it took longer than it had to take. Like it took me 1 minute to find something instead of taking 10 seconds between operations. Will it impact my data? Will it impact the results and overall conclusions of the experiment? And it those situations are even hard to explain to people who never worked in the lab. And it's a hurry with the stress, with additional stress if any errors are made. And we all humans, everyone made or makes errors in the lab. And early in my career, I had bigger stress because I had less experience. Later in my career, I had less stress because of having more experience, but I had more stress because those experiments were bigger in scale. They were more expensive. Some of them were like several months long experiments. And if you make some sorts of errors, can really impact overall results. You like work of a team of several people for several months can be ruined by my miscalculation or by my mispipetting. And it's even hard to evaluate those errors in money. You can say, it is worth of, I don't know, 20,000 of setting up the mouse work. It's worth of, I don't know, $5,000 in reagents. But if you start counting all the people's time and all the project timelines that may be broken because of those errors, you quickly start to see like these types of errors, like they're really hard to recover and they can cost a lot to the company. So yeah, and there are some errors which are not recoverable. It's just the fact of life. And of course, later in a career, I was developing different like life hacks, like how to predict in which points we can make a mistake, how to avoid the mistake just by design of like where and how we store the plastic and reagents, how we control the inventory, and we make sure that all for this experiment is in stock, and all that is in stock for important experiment is protected from other people's use. And like there are a lot of tiny details coming in play to make sure that your experiment will be done properly. And still, I believe that automation of experiments can give a lot of relief of the stress for the scientists, of just stress of the potential mispipetting, stress of potential miscalculation, and stress of just spilling something, because spills happen as well, and you can spill some, I don't know, antibody mix, which is like mixture of from 12 different tubes with antibodies, which are all expensive, and sometimes they are limited. And like if you made a spill, you kind of in front of very hard choices. Like, do I want to make a staining with concentration that is lower than the standard? But no one knows. And you can call your boss, and your boss will not know the right answer too. And I'm just making those situations up, but they can happen in the real life. So automation can help scientists to avoid human errors when doing the actual work. And I myself experienced a lot of anxiety, and I did most of my errors I did early in my career. Some of my errors I did later in my career, and it's never fun. And it is not always you have a boss who will be like totally understanding of this, and you can get into uncomfortable psychological situations, like explaining why this happens. And imagine you're a boss, imagine you're a manager, and you're managing someone's experiments. If experiment is lost because of someone's miscalculation, of someone's mispipetting, and they tell you this, like what's your feedback to give? Like, don't do it again? And like, do you need to show that you are like sad or uncomfortable or even angry or triggered somehow with this situation? Like how you handle human error, because we're all humans, and providing 100% forgiveness may be a great strategy in the beginning, but what if this happens again? And what if you as a manager are facing the tough choices of working with someone who is extremely smart, brilliant person, and understand all the underlying science, but making the mistakes because of they did not pay enough attention, or maybe They just have too much stress, and they make errors because of having too much stress. And you might end up working with people who just, I don't know, doing those operations more precisely, but sometimes they are not as creative, and they are not as brilliant with respect to some other parameters. And so it's a problem at many levels. It's a problem of someone who does execution. It's a problem of someone who is managing. And it's a problem for the company overall. Imagine a company which has hundreds of scientists. And uh, and that like big boss, if they had this stint in their career when they worked in the lab, they understand what they're facing. They're facing the potential risk of those human errors. So even automation is helping a lot. Then our technology is helping to automate some of the processes in the work of T-cell immunologists, and we are automating extremely complex, extremely long protocols, which are all error sensitive. That's number one. Number two is using less of biological sample. That's what we can solve for some niche tasks where people are looking at precise immunogenicity of some antigens. They want to know how many T cells recognize those specific tiny parts of the protein sequence, those peptides, or they can be called T cell epitopes, T cell antigens. So it's often when people need to start with very big biosamples. They need to start with, let's say, half of the blood bag, 200 milliliters of blood. And in the beginning, they need to isolate the immune cells. In case of the mouse work, people would start from taking the spleens from sacrificed animals and extracting splenocytes. When it comes to working with human, we would start with large volumes of human blood and isolating the PBMCs, peripheral blood mononuclear cells. In both situations, the outcome is extraction of large volume of cells, and I would say working with mouse spleens is easier to generate 100, 200 million of splenocytes, but when we talk about human blood, it becomes the lab work of large volume. So the blood has to be diluted in the first place. So you start from 2 milliliters of blood, you end up with 400 milliliters of diluted blood. Then this blood is used to separate PBMCs, and when we separate PBMCs by gradient centrifugation, we layer this blood on the top of a special high-density medium for separation, or we use special tubes for separation. So the point is, in this 50 milliliter tube, you can put only 2/3 of this tube of the material. Now let's do the quick calculation. You start from 400 milliliters, and you're filling up the tubes, and in each tube you can put maximum of 35 milliliters. So you need to work with, I'll just do the calculation. So 400 divided by 35, and we have 11.4, so you need to have 12 50 ml tubes. And all those tubes will be dirty with blood, and you need to do a lot of manual work. I'm not saying that it's hard to do. I did a lot of this work, but it's not something that you really like to do for several patients at once. So if you have 4 patients at once, then you have 48 50 ml tubes, and you probably need help. You don't want to do it alone. And this all like makes your overall process longer. You're moving from one tube to another, to another, to another. And many rounds of centrifugations, you start losing the cells just by centrifugation. They start to die and attach to each other. You're losing the cells because they start to attach to each other. So at least for some niche applications, if you can decrease the volume of blood, which people need to start from 20 or maybe even 100 times, let's just discuss 20 times less. If it's 20 times less blood, instead of having 400 milliliters of diluted blood, you would have 20 milliliters of diluted blood, and you use just one tube for gradient centrifugation. So one tube instead of 12, it's way easier. You can work alone with 10 patients at once instead of needing help to work with just 4 patients. So that's one of the advantages. Then whatever you need, whatever reagents you need for the sample prep, whatever plastic you need for the sample prep, all the costs go down. All the time of human work, of manual work, to do like 10 times more work, now you need 10 times less work. And this like sample sparing idea is one of the very important things. Like I, as a scientist, would love to work with 10 times smaller sample, especially with 20 times smaller sample or with 100 times smaller sample. And theoretically, we can go up to 100 times smaller samples. So that's number one thing. Then working with mice. When we work with splenocytes, we have access to like almost unlimited sample. We have a lot of sample from each individual mouse. But to obtain this sample, we need to sacrifice the mouse. And if you have multiple time points, for every time point in your experiment, which can take several months, you would need to have a separate group of animals, which will be sacrificed at this time point. And if you have, let's say, 6 groups of comparison, for each of those 6 groups, you need a separate group for separate time point. So now you start to multiply. You have 3 time points, 6 groups. Now you go up to having 18 groups of animals. And it's a lot of mice to start working with. You need a lot of your, let's say you're studying the vaccine. You need to start with a lot of synthesized vaccine material, and you need to sacrifice a lot of mice. You need to do a lot of this manual work. What the technology that we're developing can help with is just collecting the blood dynamically. You can collect at one time point, you can collect 200 microliters of blood and probably have enough material to test all of your hypotheses. So with the same group of mice, you can work with multiple time points. 3 time points, okay. You can work with mouse blood instead of mouse splenocytes. And maybe in the end of all of your experiments, you still sacrifice the mouse and work with splenocytes, but you can have multiple time points. And 200 microliters you can collect from a mouse. I believe with the current guidelines, you can collect 200 microliters every 2 weeks, every 2 or maybe 3 weeks. So this can save a lot of money for the company which orders those experiments with the CRO, and this can save a lot of hassle. And you can even use less of this synthesized like material which you use for vaccination. So yeah, and this is point number 2. Working with mice, you can remove these multiple groups of mice for every time point, and you can just literally kill less mice, which is, I believe it's just more ethical way of running experiments. So that's point number 2, but I also have point number 3. What if you need to work with some tissue that has less T cells? I'm talking about T cell studies. Our method can also, our technology can also be applied to studying different types of cells, not only T cells. And I probably will record another video about this, but with T cells. Let's imagine we want to work with mucosa, and we want to work with T cells infiltrating mucosal layers. When we extract the T cells, we can find very little number of cells. So if you need very large sample to test a lot of hypotheses, to test for a lot of T cell targets, you are in trouble. Maybe you need to pool your mouse data. You need to pool your mouse samples. So for example, you have the whole group of 5 mice. You collect the biosamples, but you cannot analyze them independently. You need to pull the sample from 5 mice at once, just to have enough of statistical power. And now on your graph, instead of having five points, 5 data points, you have 1 data point. And you report to the journal or report to your management that, yes, you have 1 data point, but it's a pooled data from 5 mice. And from the scientific standpoint, of course, you prefer to have 5 data points, and you prefer to analyze them independently. But this is in a better situation. But in a worse situation, you pull 5 mice, you pull some sample from 5 mice, and you still don't have enough cells. So you can have, like, you can test less hypotheses, you can test less strong hypothesis, less informative hypothesis, or you can just give up. You say, we don't have enough cells, and maybe in 2 time points we collected enough material, in 1 time point we didn't. So on a graph, on the dynamics graph, you just don't put the time point. You just don't put the data point. So you lost one of the time points, for example. That's one of the situations. Another situation is like you give up of the whole experimental plan. We run a pilot experiment, we try to collect enough T cells from this certain organ, and we didn't. We were unable to collect enough. For example, it's a small growing tumor. At some point, it's still too small to extract enough T cells. Or we are studying some autoimmunity, some autoimmune disease model, and we see that like there is no T cells infiltrating to some certain like tissue. And now it's time to go back to example of working with human samples. So when it's a mouse model, we can do the pilots, we can run multiple repeats, but with a clinical sample from real patients, this is often you have only 1 shot. With fresh tissue, you have 1 shot, and it can be a very tiny piece of tissue. And here, probably the best example is working with T cells attacking pancreas. So we have small piece of surgical material of pancreas. We can extract the T cells, and we want to see what are the T cell targets which are driving the autoimmune disease of diabetes type 1, when T cells are attacking the cells of pancreas, healthy cells of pancreas. And what are those drivers for this autoimmunity? What are specific T cell targets? We want to know this, but of course, there are other methods to study multiple antigen-specific T cells in the same sample. These other methods include tetramer staining, and they include, actually we can do the sequencing for T cell receptor repertoires. So I don't want to touch those methods in this video. I will discuss these as a comparator to our method in some other video. I just want to tell the story of scientists who want to do some like type of experiment. They want to study something, but they don't have enough material. And if they don't have enough material, even if they have the best finding in the world, it's impossible to run some types of experiments if you don't have the right methodology. So I hope that I touched 2 main points where our technology is solving some types of pains I had in my personal career. One type of pain, which I think that I even felt it more, is a huge stress and huge anxiety about like making human errors. And unfortunately, I was the type of person who did a lot of that. Especially early in my career, I wasn't the most attentive kid. I was the most attentive student when I ran those experiments. and I suffered a lot. I really wanted to do right experiment, but like I was unable. And I believe there are a lot of other students who suffer from this. A lot of managers and mentors who suffer from seeing that the students can run good experiments, but they just make mistakes. And the second problem is being unable to work with a sample which is too small. It's hard to obtain. I wasn't experiencing much of this because most of experiments just planned by design the way that you have enough of material. But what I experienced is the need to work with large volume of blood to extract a lot of cells to be able to run a lot of experiments. And I know personally people who have similar problems working with T cells of different antigenic specificity. So if I can help them working with less blood, I know what exact advantage in their normal day-to-day life this will bring to them. All right, that's enough for this video. Thank you for watching, and see you next time.